Okay, so now this is a different topic, and this is going to be the last video in the genetics playlist. In this one, we're going to be looking at how we know which genes have which function. So we've sequenced the genome, we know what it looks like, we know that we've got a bunch of different genes, but we don't know what any of those genes do. So we're going to have a look at how we actually figure out what those genes do. So to determine gene function, there are a few different ways that we can do it. You can either overexpress a gene by putting it under control of a strong promoter. So for example, you could put a gene under control of our California mosaic virus 35X promoter that we talked about last time, and we said it was a very strong promoter. This overexpression may alter the phenotype of the organism, so you can look at the organism and kind of quantify what the gene is doing based on looking at the organism. So say we just randomly picked a gene, we put it under control of a strong promoter, and then we see that the organism ends up having very dark fur, then we'd be able to infer that okay, the gene that we put the promoter in front of is probably responsible for a pigment that gives fur its color. Alternatively, you could do the opposite thing and knock out the gene via mutation in the promoter or coding sequence. And if we're talking about the fur color again, if the fur is white, after we knock out the gene, then we could probably infer that that's because the gene was still controlling fur color. The better way of determining gene function is through a method we call promoter-reporter fusion. This is where we link the promoter of a gene that we're studying. So, for example, the fur gene that we've been talking about, the gene that gives fur its color, to another protein sequence called a reporter. So we take the promoter of our gene of interest and we put it in front of a reporter gene, which is going to be it's going to produce a protein that we can actually assay to determine what's happening. So this is where the expression of the reporter gene is going to be controlled by the promoter of the gene we're studying. So in the organism, we're only going to see the reporter gene expressed where the gene of interest is being expressed, because they're both being controlled by the same promoter. This allows us to determine where the gene is expressed, when, and at what concentration. Reporters typically are foreign enzymes that produce products that do not interfere with cellular metabolism, because obviously it'll make it harder to study the organism if we're affecting cellular metabolism and cells are dying because of our reporter gene. So we want it to be pretty neutral and not harm the organism. And the product of the reporter gene must be detectable histochemically, and they must be localized, because obviously if we want to know where our gene of interest is being expressed by looking at where the reporter is being expressed, the reporter has to be localized so that we know where it's happening. Because if the reporter isn't localized and the protein is being spread throughout the body, it doesn't tell us anything. It just tells us that, yeah, our gene of interest is being expressed. It doesn't tell us where, so that's why it has to be localized. Easily assayed reporter proteins include beta-glucuronidase, which we shortly call GUS, luciferase, and green fluorescent protein that we shorten to TFP. In this video, we're only going to talk about beta-glucuronidase and the green fluorescent protein. We're not going to worry about luciferase here. So, beta-glucuronidase, or GUS, is an E. coli enzyme that hydrolytically cleaves a substrate called glucuronide, beta-glucuronide. It's a bit hard to say. A stable molecule that can tolerate various ionic conditions. These artificial substrates have been developed for assays, so we can determine where and what is going on. The substrate, the beta-glucuronide, is contained within this substrate called X-glue. The substrate gets broken down by our beta-glucuronidase GUS enzyme to produce an indoxyl derivative. The indoxyl derivative then gets subject to oxidative dimerization to produce a deep blue insoluble product that we can see. So all we have to really worry about is we're growing our organism in this X-glue substrate, so it contains the beta-glucuronide substrate for the enzyme to break down. The enzyme breaks down that breaks down that substrate, and we get a blue color because of it. We don't really have to worry about stuff like what an indoxyl derivative is or what oxidative dimerization is. We just know the enzyme breaks down the substrate and it makes the organism blue where the GUS is being expressed. But GUS has a key limitation as a reporter gene, because in order for us to view the deep blue color, the organism must be fixed, which requires us to kill the organism. So in vivo studies are not possible, and we're only able to view where genes are being expressed at specific times when we decide to kill the organism and view it. An example of GUS being used was when the beta-glucuronidase, the GUS enzyme coding sequence, was added to the end of a promoter that promotes a gene called extra plant extension cell wall protein in tobacco. And then we had a look at what parts of these fixed tobacco plants were showing the deep blue color. So this is where this extra plant extension cell wall protein gene was actually being expressed, because they're both being regulated by the same promoter, and it was found that this gene was being expressed in the in the plant roots and in the kind of apical meristem. That's not the exact picture from the study, I couldn't find it, but you can see there how GUS is giving us this dark blue color in the roots and in the cotyledon leaves, which would indicate that this is where the gene of interest is being expressed. The other reported gene we use is green fluorescent protein. It can be used to study organisms in vivo, so it can study living organisms. We originally found this gene in the jellyfish Aquaria aquaria, and in these jellyfish, TFP produces blue-green luminescence. In the jellyfish, when they're touched or stressed, they release calcium 2+ ions that bind to aquarin, causing it to causing it to emit blue light at 460 nanometer wavelength. This blue light then excites the TFP proteins, causing them to shine a fluorescent green at 509 nanometer wavelength. Using the TFP reported gene in association with the promoter of our gene of interest means that we can shine a blue light at 460 nanometers on our organism that we're studying, and this living organism will respond by shining fluorescent green wherever these genes are being expressed. So if you look at the mouse on the right, where the mouse is shining green, that's where its genes are being expressed. We have a bit more to cover on a process called promoter trapping. I was hoping to get it done at the end of this one, but there's a bit much to include at the end of this, so I'll do a short video in a minute on it.